The central dogma. It's the code of life. You gotta live by a code in this life. Mine is in my DNA. What's yours? No, really. What's your code?
The development of the structural DNA model.
We all know what DNA is, or do we? In this section, we'll learn what we know and how we know it. That is, we'll take a look at the basic structure of DNA and learn about the researchers who developed this model. Hit it!
So I know DNA is a nucleic acid in the nucleus that contains genetic information like instructions for how an organism will develop. But what's it made out of?
Glad you asked. Let's go back to the 1940s. In the 1940s, a biochemist named Erwin Chargaff developed techniques to analyze the chemical composition of DNA, that is, to figure out what it's made out of.
So, when Chargaff came on the scene, it was already known that one of the main components of DNA was a nitrogen-containing base. There are actually 4 of these bases: adenine, or A, thymine, or T, guanine, or G, and cytosine, or C.
Enter Chargaff. He figured out that in any species, the amount of adenine always equals the amount of thymine. So A always equals T. And that the amount of cytosine always equals the amount of guanine, or C always equals G. Chargaff found that different species have different amounts of these bases, but in any species, A always equals T and C always equals G. Always, always. Chargaff was a real stickler for the rules, and these equations are known as Chargaff's rules.
Okay, so Chargaff noticed that A equals T and C equals G. But why? Why? Where's this all going?
This is bringing us to an understanding of the structure of DNA, which is super important.
Let's move along the DNA timeline and see what was discovered next. Next up, we have Rosalind Franklin in the year 1951. Franklin used technology to take these cool photos of DNA called X-ray diffraction photos. These photos were basically projections of the patterns that DNA molecules make. By taking one look at these patterns, some clever scientists were able to figure out the three-dimensional shape of DNA. And so, with Franklin's photos, the shape of DNA was finally figured out. What was it, you ask? Well, the shape of DNA turned out to be, drumroll please, a helix. But not just a helix, a double helix. Wait, what's a double helix?
It looks like this. This is a helix. You can think of it like a corkscrew. And here's a double helix. You know, there's just like 2.
So who were these guys who were able to look at Rosalind Franklin's X-ray diffraction photos and decide, using their super mathematical skills, that the shape of DNA is a double helix? Glad you asked. These guys were the famous Watson and Crick, the first ones to actually construct the DNA model, and the next guys we'll study on this DNA timeline.
Watson and Crick! What must it have felt like to discover the very structure of DNA? Must have spent a lot of time building with blocks as children. Childhood. I was never a child. I sprung from my mother's womb fully formed.
So how exactly did Watson and Crick build their model of DNA? Well, let's consider what they knew. They knew the shape was a double helix. They also knew that this helix is made up of the nitrogenous bases, which we already learned about, A, T, C, and G, and that it is also made up of a pentose sugar called deoxyribose, as well as a phosphate group. Don't worry too much about these terms. Let's just concentrate on what this meant for the structure.
Watson and Crick also knew that Rosalind Franklin had concluded in her research that the sugar and phosphate stuff formed the backbone, or the outside of the double helix, and that the bases were on the inside. Think about it like a ladder. Here's the sugar-phosphate backbones. They're like the rails. And here are the bases. They're like the rungs. Each chain of DNA has 2 strands, or rails, with pairs of bases climbing up between them, like rungs.
Now, even though Chargaff said that A always equals T and C always equals G, Watson and Crick weren't sure until they were sure. They tried all different ways of pairing the bases, and here's how they figured out why A always goes with T and C always goes with G. Listen up.
First of all, A and G are both a type of nitrogenous base called purines, while C and T are pyrimidines. Now, the purines have 2 rings and are wider than the pyrimidines, which have 1 ring. Since the DNA ladder is the same size all the way down, meaning all the rungs are the same length, otherwise it would be a weird wonky ladder, it makes sense that each rung is made of pyrimidines and a purine. That is, 1 large and 1 small. Never 2 larges or 2 smalls together.
So why not A with C and G with T? That would still be a big with a small. Why does it have to be A with T and C with G? Well, there's one more piece to this puzzle. Watson and Crick discovered that A and T are sort of like soulmates, since they're able to form hydrogen bonds with each other. Same with C and G. They're partners. And that's the way it is. A with T and C with G, consistent with Chargaff's rules. Watson and Crick created the shape of a ladder with A across from T and C across from G's. Then they twisted the ladder to form a helix. Voila!
So there you have it. The basics on the structure of the DNA model and the researchers who contributed to its development.
The central dogma.
Central what?
The central dogma is the process in which DNA, by the way of RNA, is used in the production of proteins. DNA's main role is to provide the crucial instructions needed for protein synthesis. And why is this so important? Well, it is the design behind every organism on the planet, after all. And the central dogma tells us step by step how it all goes down.
By the way, remember that old double helix shape? The central dogma is going to help us understand why that shape is so important to what DNA does. When you think about it, that structure is probably one of the most important shapes on the planet. Yeah.
Madame Vocabulary here. Travelers, you are entering the mysterious realm of DNA code. Here is some vocabulary to light your way. Don't worry about memorizing them right away, of course. Just get familiar.
DNA, or deoxyribonucleic acid, is a molecule that is shaped as a double helix and that has the ability to replicate itself in order to pass along genetic information to other parts of the cell for protein synthesis. RNA, or ribonucleic acid, is a nucleic acid made up of a single strand that is a key player in the process of protein synthesis. Transcription is the process by which DNA is copied into messenger RNA. It occurs in the nucleus. Translation is the process by which messenger RNA is interpreted into the language of amino acids to produce a chain or sequence of instructions for protein synthesis. A codon is a group of 3 nucleotides, like a word that stands for a particular amino acid or a start or stop signal in protein synthesis. Nucleotides are the subunits that make up DNA and RNA molecules. Each nucleotide is made up of a nitrogenous base, such as adenine, thymine, cytosine, guanine, or uracil. Ribosomes are the little machines in the cell that make proteins from amino acids. They are located in the cytoplasm.
Welcome to the path of the central dogma. This is going to be an eye-opening journey. First, we're going to learn about how DNA makes copies of itself. Then, we're going to learn about how DNA is transcribed into the stuff called RNA. And finally, we're going to see translation and the cracking of the DNA code. That's a hefty itinerary. First stop, DNA replication.
DNA replication. Watson and Crick, those famous guys who built the double helix DNA model, also wrote an important paper talking about DNA replication. They ended the paper with this intriguing sentence. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for genetic material. A copying mechanism for genetic material. Sounds interesting. Sounds sort of familiar. In this section, we're going to find out about this mysterious copying mechanism.
Once we have the proper footing, the copying mechanism of DNA is really simple to understand. It just makes so much darn sense. Okay, so we've got our twisted ladder with its 2 sugar phosphate backbone rails and these rungs of paired bases.
And we know that each of these base pairs are like soulmates. A and T are meant for each other. They know everything about each other. They complement each other. Same with C and G. In fact, these guys know so much about each other that each one even contains the information of how to reconstruct the other. Wow.
So they're soulmates, but get this. They don't have any attachment issues because even if they get separated, they know how to make a copy of their mate.
So Watson and Crick realized this, and they realized it was the perfect setup for making copies. They figured it was possible to unzip the two strands of DNA, basically tear the latter apart straight up the middle, separating all the pairs, and that these lonely lovers would then be able to use their information to create a copy of their missing mates. Then the backbones would bond together and you'd get two identical strands. This process of duplication is called semi-conservative replication. This is the process DNA follows to duplicate itself.
It's just so simple, so elegant. Like my wife's wardrobe of stretch pants and pearls. She loves to exercise. You see, the function of DNA is directly related to its structure. That is the beautiful thing that Watson and Crick realized. Once they saw the structure, they knew it could duplicate itself. Once I saw my wife's face, I knew she could make excellent spaghetti.
Next stop along the central dogma, RNA and transcription. Let's go.
So we just learned about how DNA duplicates itself, but why does it duplicate itself? Good question. Let's take a look at the bigger picture. We need DNA to make proteins, right?
Well, when you think about it, it's kind of weird because DNA is located inside the nucleus of the cell and proteins are made out here, in the cytoplasm, on the ribosomes. Seems like we're missing a link.
We are missing a link, and it's called RNA. More specifically, a particular type of RNA called mRNA, or messenger RNA, because it serves as a messenger, delivering information from the nucleus to the cytoplasm.
Golly, DNA is such a diva. Why doesn't it just deliver the information itself? Why get this RNA involved?
Well, think about it. DNA is the long-term storage of precious genetic information. If it leaves the nucleus and anything happens to it, that would be bad for the organism. It would end up missing a nose or something.
But how does DNA get the information to the RNA?
Glad you asked. That brings us to our next topic of discussion, transcription. DNA is transcribed into RNA. We use the term transcription because we're only making a copy of the genetic code, just writing the same information on a different piece of paper.
Pretend the DNA is an original Spider-Man comic. Episode number 1, signed by the author. It's precious with a capital P and has to stay inside a museum just like the DNA stays inside the nucleus. The RNA, meanwhile, which is single-stranded and not a double helix, is just like any copy of the Spider-Man comic that you can find in the bookstore. It's got the same information, but it's way cheaper and easier to come by.
Hey guys, remember semi-conservative DNA replication? How the DNA strands unzip from each other and the nitrogen bases make copies of their soulmates? Well, you guessed it. The transcription of DNA into RNA goes down in nearly the same way as semi-conservative DNA replication. So, if you understand that, you've also got RNA synthesis down. Let's take a look at how that RNA copy is made.
The helix of the DNA is unzipped, and the bases of RNA line up with the bases of DNA and form hydrogen bonds with them. Now, you'll notice that the RNA here is missing the thymine, or T base, and it's got this U base, called uracil, instead. No problem. Instead of the A's in DNA pairing with the T, they pair with these U's. Meanwhile, the T's in the DNA still get to pair with the A's in the RNA, just like usual, and the C's and the G's pair with each other, just like usual. So everyone's more or less happy. Remember, RNA is composed of U, A, G, and C, while DNA is A, T, C, and G.
Now let's finish up this transcription. After the initial copy is made, it has to get processed a bit more before it is ready to leave the nucleus and travel to the cytoplasm as a mature mRNA.
One important part of this processing is the removal of introns. Introns are non-coded segments of the RNA. These are cut out and the ends pasted back together so that the strand forms one continuous code. This is called RNA splicing.
The finished mRNA looks like this. As you can see, it has several regions. The largest section in the middle there is the coding region. This is the area that contains the information that will be translated. The areas to the left and the right of it are untranslated regions, or UTRs. Remember, the coding region is the part that actually gets translated.
The finished mRNA is now ready to leave the nucleus and go out into the cytoplasm, where the ribosomes are, and where the information it carries will be used to make a protein.
Before we move on to the next step in the central dogma translation, let's talk for a second about different types of RNA. There are 3 major types of RNA. They're all made in the nucleus, and they all help in protein synthesis. One, mRNA, is, as you know, the messenger used to make proteins. Another, rRNA, or ribosomal RNA, is found in the ribosomes and is involved in translating the mRNA into amino acids. The third type of RNA is tRNA, or transfer RNA, which is like the forklift that delivers an amino acid to the ribosome, where it then follows the directions provided by the mRNA to make proteins. So, RNA comes in various forms, and these various forms are used throughout the process of going from DNA to protein. Don't worry, we'll talk more about these different RNAs in a few minutes when we break this all down.
Next and last stop, translation and the deciphering of the DNA code.
Yes. I remember my days working in service of the Pajit Dutsma tribe, high in the mountains of West Walawatsu. They taught me their language, and whenever a traveler from foreign lands came to the village, I would teach him the word for leave in Walawatsuyan, and then the villagers would chase the traveler out with smelly banana leaves.
Translation. Once the mRNA is in the cytoplasm, it must be translated to make a protein. Think about it this way. The DNA and the RNA speak the same language, right? The language of nucleic acids. But protein is made of amino acids, and that's a whole different language. In order to make a protein, the amino acids need the info from the DNA, and they obviously need it provided to them in their own language. So that's right. It's gotta get translated.
Okay, that makes sense, but how does this translation occur? Well, first, a ribosome, which is the machine that makes the protein, slides down the untranslated region until it finds a start codon.
Translation has everything to do with these things called codons, which are sort of like words. Each word is made up of 3 letters or bases. You know, A, T, C, G, or U.
Each codon stands for an amino acid. A sequence, or sentence, of amino acids makes up a protein. So the mRNA is read by the ribosome in terms of these 3-letter codons, and these are translated into the language of amino acids.
Let's repeat that. The ribosome reads the mRNA. It reads 3 letters at a time. Each 3-letter codon, or word, corresponds to 1 amino acid. Okay, but how do we know which codon corresponds to which amino acid?
That's easy. Just memorize this table. I'm kidding. Don't memorize this table. Let's just learn how to use it. This table is the key to deciphering the DNA code. It's sort of like a French to English dictionary, except that instead of translating French to English, we're translating nucleic acid to amino acid. Here's how to use it. We start with the first letter of the 3-letter codon and find it on the left side of the table. Then go to the second letter of the codon and find it on the top of the chart. Then find the third letter of the codon on the right side. Then find the amino acid on the chart where they all meet.
Let's try it. Say you have the codon UUU. You find these 3 letters on each side of the chart, then find the point where they meet, and you end up at the amino acid PHE, which stands for phenylalanine. The codon GCA corresponds to ALA, or alanine. Wow, what a handy decoder. That makes translation so easy.
This table is the same for pretty much all life on Earth, from amoebas to zebras. What does that mean? It means that the genetic code is evidence for the shared evolutionary ancestry of all living things. That's right. This code makes it clear that we all came from the same stuff. Whoa.
Now, although you don't ever need to memorize that whole DNA decoder table, there is one thing that you do need to memorize. It's just this one codon, AUG. AUG is the start codon, and it's a handy tool. Here's why. If you need to translate a long strand of mRNA, all you have to do to know where the sentence of coding starts is skim along the untranslated region of the mRNA until you find the start codon AUG. And then, and only then, do you start translating. AUG corresponds to the amino acid methionine, which is the first amino acid of every protein.
So we've got a start signal. Maybe you're wondering, what about a stop signal? Notice on the table that there are 3 codons that don't refer to any amino acid. You guessed it. Those are the stop signals.
So now that we're getting to know this table, there's just one more thing that you should notice about it. Notice that many of these amino acids have more than one code that stands for them. Think about it like a dictionary. The words stinky, foul, and funky all mean the same thing, smelly. Likewise, in the chart, you can have multiple codons that lead to the same amino acid. As you can see, there are 64 triplet codons, but only 20 common amino acids, meaning that each amino acid must have more than one codon, or word, that corresponds to it.
Okay, that's really cool, but can we finish this process already? I mean, I want to see the finished product, the protein.
Let's do it. So we're on the ribosomes, remember? That's where translation occurs. The ribosome is made up of rRNA. That's ribosomal RNA, and protein. The ribosome is the machine that bonds individual amino acids together to make a protein, and the rRNA is the interpreter of the nucleic acid codons. But we're missing a step here. How do the corresponding amino acids get from the sea of cytoplasm onto the ribosome?
The answer to that is tRNA, or transfer RNA. tRNA is like some big dude with a forklift that picks up the amino acids in the cytoplasm and transfers it into the ribosomes. In reality, tRNAs are small RNA structures with a 3-base anticodon at one end that attaches to a corresponding 3-base codon of an mRNA. Meanwhile, the other end of the tRNA links to the specific amino acid that the codon stands for.
Here's a tRNA. It's got 3 hairpin loops. The anticodon is at the top of the second loop. The anticodon pairs with the mRNA's codon according to our usual base pairing rules, A with U and C with G.
So, we're nearing the end of our protein-making process here, coming to the end of the central dogma path. Let's talk a bit about the resulting proteins. One thing to point out is that because mRNAs are all different in length and sequence, proteins translated from these mRNAs are going to end up different in length and sequence.
Say that again?
We're talking about sequence. Let's go back for a second to that DNA molecule in the nucleus where this all got started. The genetic information it contains is dependent on the order or sequence of the nucleotides. So when the mRNA delivers this information to the ribosomes, it must deliver it in its proper order so that the amino acids get strung together in a particular order. This order determines the physical properties of the protein and therefore the proper expression of the genetic information.
Remember how we were saying that you can think about the sequence of amino acids that make up a protein like a sentence? Well, depending on the order of the words, the sentence is going to end up differently. For example, look at these 2 sentences. I wish for a rainbow-colored jellyfish. I wish fish colored jelly for a rainbow. They're both made of the same words, but when the order changes, the relationships between the words change, and so does the overall meaning. Likewise, depending on the order of the amino acids, the protein is going to turn out differently.
Woohoo! I'd say we've doggone got it.
Gene regulation.
Did you know that all body or somatic cells have identical DNA? That means that all of your cells could end up anywhere in your body and they know what to do to work in that area. That is, they'd have the information to tell them what to do. Think of it like a play in which all the actors not only know their own lines, they know everyone's lines. So anyone could play any part, just like any cell has the information to work in the eye or the heart or the kidney, anywhere.
Now, little cell, remember, you can be anything you want to be when you grow up.
Okay, but if your cells are all identical, doesn't that mean you would just be a big blob of sameness?
Well, yes, you would be a big blob of sameness if it weren't for gene regulation.
All your genes are regulated in terms of what knowledge or information they're able to express. Therefore, the genes of the cell that's in your leg won't express proteins that are needed only in the eye. Otherwise, you'd have an eyeball growing out of your leg.
But I could have been anything. A doctor, a lawyer, a mailman.
Sorry, little cell, but gene regulation is important. Why is it so important? Well, clearly, so we're not all just blobs of sameness. But also, if anything goes wrong with a gene expression, this can lead to serious imbalances or diseases in the body, even cancer.
Let's be clear here and review this simple, important fact of gene regulation.
Specialization of cells in multicellular organisms is due to different modes of gene expression and not due to differences in the genes themselves.
Got it? Good.
DNA mutations.
Mutations are changes in the DNA sequence.
Mutations. DNA getting messed up, leading to all sorts of problems. Or to something wonderful, perhaps.
All right, mutations. Let's talk about it. Remember our 4 bases in the DNA strand, A, T, C, and G? Well, a mutation occurs when one of these letters gets changed to another letter.
Before we go on, let's take a second to get something straight. The truth is, it's more correct to say that a mutation occurs from a changed nucleotide rather than a changed base. Remember that a nucleotide is simply a unit of DNA or RNA that includes a base, along with a sugar and phosphate group that form the backbone of DNA. Don't worry about these specifics too much right now, just be aware of them.
You may be wondering, how does this happen?
Well, mutations are caused when one of the nitrogenous bases gets messed with.
Ouch! That can't be good.
So what's the effect of a little base change in the DNA?
Well, let's consult the central dogma. Remember, the central dogma is the process by which an original DNA code is transcribed into mRNA, which is then translated into protein.
Remember, each protein is made by a sequence or sentence of codons. Each codon, like a word made of 3 bases or letters, refers to a specific amino acid, and a protein is a sequence of amino acids. Here's a normal amino acid sequence. The first line shows the DNA sequence, and the next line shows the transcribed RNA. The third line is the amino acid sequence resulting from translation of the RNA. Normal, normal, normal. Now, let's see what happens when the DNA gets mutated. Whoa! Now, let's say this cytosine here, C, gets changed to an adenine, A. Considering how precise those bases are about who they get paired up with, you know, A with U and C with G, this could get interesting. Let's see what effect this has on the protein. First, watch what happens in transcription. In this case, the imposter A gets transcribed into a U. So, our mRNA codon becomes UCU, which translates to serine, not alanine. This type of mutation is called a missense mutation, because in that mutant codon, there is a mistake protein being translated. So a missense mutation is when just one base has been changed, resulting in a codon for a different amino acid. Well, at least the codon is referring to an amino acid, so it still makes sense. It's just the wrong amino acid.
Like if I said the sky is purple. That would be wrong, but it would still be a complete sentence, and purple is still a color. It's just the wrong information.
Missense mutations can be the cause for certain diseases, such as sickle cell disease.
There's another type of mutation, a nonsense mutation. This is when the codon for the amino acid gets changed into a stop codon, which causes the translation to get stopped before it's been completed. Like if I said, the sky is, and then just stopped. That makes no sense. Get it? Nonsense mutations can cause genetic disorders.
A terrible, terrible situation. How long have I been saying? Grammar, 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 punctuation, proper spelling. How else do you expect to communicate properly in this world? What are we talking about?
Now let's look at another type of mutation. This one, called a frame shift mutation, gets pretty messy. Watch this. Say we insert an extra thymine in the DNA here. What effect will this have on the outcome of the translation process?
Well, all of the codons are always groups of 3 nucleotides each, right? When you throw in an extra nucleotide or letter, it's going to get grouped into a triplet, pushing one of the letters from that triplet into the next, pushing one of the letters from that triplet into the next, and so on, jumbling up all the triplets.
Say I had the sentence, "Fat cat ate mud," and for some reason, I had a computer that only allowed 3 letters per word. If I added an S to the end of fat, the sentence wouldn't just read, "Fat's cats ate mud." Instead, the computer would focus on having 3-letter words and changing it to fat, ska, tat, emu, d. Complete gibberish. You can see how disastrous frame shift mutations can be.
They can also occur when a nucleotide gets deleted, and things end up getting just as confused, leading to missense and nonsense translations all across the board.
One example of a disease caused by a frame shift mutation is Tay-Sachs.
Finally, we have something called a synonymous substitution. This is sometimes called a silent mutation because, although a base in the DNA gets changed, this change has no effect on the final protein.
Think about it. Remember how there are multiple codons that translate to the same amino acid? You could say they're like synonyms, 2 different words that have the same definition. So, when a mutation causes for a synonymous codon, it translates to the same amino acid as it would have in the first place. So, these synonymous codons are sort of like a built-in safeguard against the disastrous effects of mutation. Neat!
That's all for now, folks. Beware of mutant DNA. Ha 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 ha.
DNA technologies.
Now that we understand how DNA works, let's talk about how we work with DNA. That is, technology scientists have developed to study DNA.
Traveling to the future today, travelers? The future is now. Here is some vocabulary to brighten your day. Please don't trip over the long words or you'll frighten my donkey.
Cloning is when a bunch of copies of a piece of DNA are made in a lab. Recombinant DNA is when DNA from 2 different organisms, often different species, is combined in a lab. Endonucleases are enzymes with sticky ends that cut through a DNA strand so that another piece of DNA can be inserted into the strand. Gel electrophoresis is a commonly used technique used in studying DNA, in which a gel is used as a strainer to separate nucleic acids or proteins according to certain physical properties, such as size or electrical charge. Ligation is when the ends of 2 DNA molecules are covalently linked using a ligase, which is an enzyme that activates the formation of the covalent bonds. A vector is a vehicle that can carry foreign DNA into a cell and replicate inside the cell. Transformation is when foreign DNA is brought into a cell, causing a change in genotype and phenotype.
Molecular cloning is when researchers take one little sequence of DNA, throw it in a test tube, and make a bunch of copies to study. In this section, we're going to take a look at the steps scientists take to clone DNA for research.
3 steps involved in molecular cloning are DNA fragmentation, ligation, and transformation.
Alright, so we're going to clone some DNA. Let's walk through this one step at a time. First, let's discuss how to fragment DNA. Fragmenting just means we're breaking it up into pieces or chunks.
Although there are multiple ways to fragment DNA, today we're going to talk about one of these ways called digestion with restriction endonucleases.
What?
Digestion with restriction endonucleases. Let's break down the word endonuclease.
Ase, here at the end, means that this is an enzyme that cuts something. What does it cut? Nuclei here means that it cuts nucleic acids, and this first part, endo, means that it cuts within. So an endonuclease is an enzyme that cuts through the middle of the DNA.
So we take an endonuclease and use it to cut the DNA. The special enzymes that we use to do this are called restriction endonucleases. That just means that they cut only at a specific area of DNA, and they leave the ends of the freshly cut DNA all sticky. So now another complementary strand of DNA that has been cut along the same area can be stuck to it. and they can be linked together, like links in a chain.
The next thing you need to do after fragmenting your DNA is to separate out the part of it you want to study. The easiest way to do this is gel electrophoresis.
When I was a struggling graduate student, I once tried to substitute the beautician's learned touch with an application of gel electrophoresis, resulting in an uncombable, tangled mess, to which my bald brother belly laughed big time.
Gel electrophoresis is sort of like running your DNA through a strainer made of gel to separate the different pieces based on their sizes. The gel has the consistency of firm jello.
It works like this. DNA has a negative charge from all those phosphate bonds in the backbone. So when you run an electric current through the gel, the negatively charged DNA is drawn towards the positive pole on one end of the gel. Small pieces of the DNA are able to zip through all the little pores fast, while big pieces move more slowly. As a result, the pieces of DNA are separated into bands based on size.
After you have a bunch of fragments of interesting DNA, you need to link them to a vector, which is a vehicle that can transport this DNA into a cell. A vector is just another small piece of DNA that hasn't been manipulated. The foreign stuff sort of gets hidden inside it so it can get into the cell. This process of linking, which utilizes covalent bonds, is called ligation. Ligation is when an enzyme called ligase is used to make the covalent bonds form.
After ligation, this freakish bit of DNA you've created is injected into bacteria. Once the foreign DNA is inside a bacterium, it will ride along with the bacterium like a parasite, replicating each time the bacterium replicates, just like it's an extra chromosome. So, this foreign DNA has not only been introduced in the bacteria, but it is now replicating and swapping info with the bacteria's DNA. The resulting mixture of DNA is called recombinant DNA.
You must be wondering what happens with the introduction of this foreign DNA into the cell's replication process. Well, it changes the genetic makeup of the bacteria, as well as its phenotype, or the way it looks. This is called a transformation. Totally sci-fi and totally useful for research in genetic engineering.
And that's how you clone some DNA. Remember, don't try this at home.
Thanks for watching our show. Goodbye and good luck.